This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 380, recorded on March 11th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Boston, Massachusetts, Rich Condit.
Howdy, Vincent. How are you?
You know, I had to say from Boston because that's where you physically are, right?
That's correct. I am physically in Boston, where the temperature is 58 degrees and what's that, like 14 Celsius and a clear blue sky. It's I think that's unseasonably warm for Boston.
It is.
Others would know better than I, but that's cool.
On Monday here, it was freezing. I wore gloves and a heavy coat and a scarf. And today it's 16 Celsius. Yesterday it went up to 25 Celsius here.
Whoa.
Pretty warm.
Yeah, that's right. When the day we were driving up, it was like 80 Fahrenheit around the New York area as we drove by. Yep.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And Rich, I'm facing east and waving to you right now.
Oh, okay.
You're right across the state.
I don't know which way I'm facing, but I'll have to get out my compass on my my phone. But I'm waving to you. I'm waving at the window, figuring that's probably better than waving at the wall.
That's close enough. And it's pretty much the same weather here. It's nice, just a couple of clouds in the sky and 59 Fahrenheit for those who want Fahrenheit. It is a little breezy, but that's because the front just blew through.
So it turns out I'm waving south, so I got a ricochet off the tree there, and
okay.
I'll get to
Microwaves.
Waves do reflect.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How are you?
Great. It's 51 degrees Fahrenheit, 11 Celsius, sunny with wispy clouds, and I biked in for the first time in 2016.
Cool.
Is that,
you try to do that whenever you can now from now on, right?
Yep. Mm-hmm.
That's pretty neat. Now, how long does it take you?
About 10 minutes.
I think
Not a very long commute.
I think Rich used to bike in, right?
Absolutely, I still do. When I go in, which is not infrequently.
Yeah, you go into interview and
go to seminars and stuff like that. Schmooze, schmooze.
You go to seminars, yeah.
I go. There's a journal club I like to attend. There's a clinical case presentation that I like to attend, and there's a couple of different seminars I like to go to. Yeah,
nice. This episode of TWiV is sponsored by the Auger Art Contest. This is an event sponsored by the American Society for Microbiology. It's the second one. They had one last year, and it was pretty cool. The winners were just amazing. You basically make something on an agar plate, so you kind of need to have access to agar plates and to be able. Most people spread bacteria or fungi on the plates in artistic ways. They're pretty cool. And this is the second one. And what you do is you submit a photo of your original agar-based art and a short description of it. And the deadline is Friday, May 6th, 2016. You have a lot of time.
Right. But you don't submit the actual plate.
No, you submit a photo of it, yes. And so you have a lot of time. If you have a slow grower, you can start now. Winners and select runners-up or runner-ups. What would it be, Alan? Runner-up?
I think it's runners-up.
Yeah.
I would say runners-up. It's like fathers-in-law,
Right.
Yeah.
Yep, I agree.
I agree with that, too. I'm going to change it here. We'll be showcased in a gallery at the ASM Microbe meeting, which will happen in Boston this June. And I will be there, and Alan will be there.
Yeah, I'll have to check that out.
We will. All winners will also get a free ASM book of their choosing. I'm sure you'll all want Principles of Virology.
Unless you already have it.
Right.
That's right. If you do have it, you can get another one. And you can run a contest and give it away. Woohoo! For full contest guidelines and terms and conditions, and you have to sign a 30-page, what do they call those for software? The user thing.
User agreement or end user EULA.
EULA. I'm just kidding. You don't have to sign EULA. Go to bit.ly/agarart 2016. That's all one word. AGARART 2016. And let's see what you can come up with. I was on the judging panel last year. It was a lot of fun.
Oh, that would be fun.
And I think I was on it this year as well. I am on it, I think. Lots of fun.
I'm glad they're continuing that.
Yeah, it was a big response and lots of interest all over the place. So that's a clear winner.
That augurs well for its future.
Yeah. And we have a show title. That's okay. All right, we have some follow-up from Jared, who writes, "Hi, just a couple of brief comments on the spread of rabies within bat populations. Doesn't the TWiV team know about blood sharing? Reference attached. Also, I would like to second the call for more episodes on plant viruses. Personally, I would love to know if there is anything known on the interaction of orchid and symbiotic fungi viruses." All right, well, we don't have orchid and fungi today, but we do have a plant virus paper, Jared, okay?
And thanks to you, Jared, we now know about blood sharing.
We do. We didn't know.
This is interesting.
It's a paper published in 1984 in Nature called Reciprocal Food Sharing in the Vampire Bat. It's a fellow in La Jolla at UCSD.
And they apparently puke food into each other's mouths to...
Mm-hmm.
Yeah. And their food, of course, is blood.
Highly appealing behavior.
Yes.
Thank you, Jared. We don't know everything. In fact, we know very little, and we're always being educated by our listeners. Why don't you take the next one, Alan?
Sure. Let's see.
Severin, right?
Severin?
No idea. Ask Kathy.
I think Sewern, yeah.
Severin?
Sewern, yeah.
It looks almost Welsh.
So we're in all this. So we're in all this together, right?
Mm-hmm.
Yes.
Oh.
Sorry.
Writes, "Dear TWiVeroos, greetings from a partly sunny Brisbane, where it's 24°C and 83% relative humidity on this 8th day of autumn, in quotes. Just a quick comment on the recent discussion on bat transmission of rabies. Bat lyssavirus, a close rhabdovirus relative of rabies, is capable of causing rabies-like disease in humans through bites as well as scratches, presumably from claws covered with residual saliva. As noted by the eminent podcast panel, bats are highly social animals which live in densely packed colonies, so transmission between bats could potentially occur through intentional or accidental scratching, as the individual bats are jostling for position, fighting, mating, etc. And then P.S. Please be kind to Dickson. Musings and speculation form important parts of the lateral thinking and discussion process and can lead to novel concepts and discoveries." And Severin is in the Queensland Pediatric Infectious Diseases Laboratory at University of Queensland.
I totally agree that lateral thinking is important. I couldn't agree more with you. However, Dickson is not doing that. He's just going over stuff that we've already covered in which we know the answer. Okay, I don't need to go into it.
Well, sometimes he is. And I think it's a little bit frustrating because when he's not here, he doesn't listen to the podcast, so he doesn't know some of the things we've already covered.
But I totally agree with this whole thing. Sure, musing and speculation, we do it all the time, absolutely. But, I'm too close to Dickson now. It's I shouldn't get annoyed with him. I don't get annoyed with anybody else, as you notice, here on the panel. So, hey, I discovered who the fake Dickson is.
Who? Oh, no kidding.
He's a former student of my virology class.
Oh.
came by.
Now, is that the one? There are two fake Dicksons. Is that the one? Is that the one who's on as fake Dickson Despommier, or the one who's on as vertical or VF doc?
It's called. He's the fake Despommier. Yeah.
Fake Dickson Despommier. Okay.
Yeah, and he came by after class this week, and I said, "Are you the fake Despommier?" He said, "Yeah."
How did you guess?
Well, I had given a lecture in another course last semester, an immunology course, and he was in the audience. And as I started lecturing, he held up a sign saying, be nice to Dickson, which I posted somewhere. I suspected it was him. And then he showed up, so I asked him. Cool. Now, this past week, some of you may be interested in this continuing FBI Apple controversy, which I picked a couple weeks ago. So I'm reading a lot about it. And I think it was last week, Gary Fagan, the chief deputy DA for San Bernardino County, said on this iPhone that they want to open up. The DA, Michael Ramos, made the outrageous statement that Farouk's phone might contain a, quote, lying dormant cyber pathogen. And I thought that was funny. So I tweeted, first they steal viruses, and then now they want to steal pathogens. And of course, Alan then tweeted, I think they took worms first.
Yes. From
TWiP. And of course, he totally made this up. So I'm going to post a link to Michael Zdarsky's blog of things, which I picked a couple weeks ago, which is really a nice analysis of it. He said basically this doesn't actually exist in any computer science, let alone in information security. Isn't it funny? A cyber pathogen. They actually knew what the word pathogen was.
Yes.
Whereas a pathogen is something that causes disease, and of course you could co-opt it for digital issues, but no one has until now.
And a lying dormant cyber pathogen is grammatically incorrect as well as computationally.
So this is getting very nasty. This whole thing. Yesterday, so my pick a couple of weeks ago was Apple's motion to what is the word they use? Not dismiss, but there's another word which I
vacate.
Motion to vacate.
Right.
And now the Justice Department came back with this scathing personal thing, which like has totally shocked Apple, and it's really weird.
So your son's into cybersecurity, right?
He is. I should talk to him about this. I want to know what he feels about it. He's coming home next week for break. I will ask him. I have not disturbed. You know, the thing is, if I text him, he wouldn't answer for 5 days, and then so I'll catch him when he comes back. If this were a cybersecurity show, I could have him on, but it's not.
So I want to know what this picture is on the face of Zdarsky's blog of things, with the houses on the water.
Oh, so he's a photographer, and that was one of his ones from 2015. If you go back far enough in his blogs, he's got a post right at the 2015-2016 boundary that's his pics of his favorite photos. And
his photos are amazing.
Yes. Yeah, he describes where they are and stuff.
Is this one Amsterdam? Is this Amsterdam?
It looks Dutch.
I'm thinking it's either Amsterdam or Curacao, because there's a scene very much like this in Curacao, right downtown. There's, but I think this is probably Amsterdam.
Yeah, it's a beautiful image.
It's lovely.
Yeah. All right, now we have for your listening pleasure the Zika Report.
We need some theme music for this.
Oh yeah, what kind of music would you have? Like a scary music?
The thing that pops to my mind is the Twilight Zone theme.
Yeah, but it's probably copyrighted. I can't just put it.
Yeah.
In.
Well, you just sing it like that. Just take that and loop it.
Yeah, people want to hear me off key. Anyway, we have a lot of things here, but we won't take a long time.
We should have something with more of a samba beat, maybe.
Why would you do that? Oh, yeah, I guess
Brazil?
so. For Brazil, yeah.
South
All right. First thing, the papers are pouring out.
Yes.
And that's actually relevant to this first one, which is Zika virus detection in urine from patients with Guillain-Barre syndrome on Martinique, January 2016, which was published in Eurosurveillance. So we're seeing more of Guillain-Barre, which is a peripheral myelitis, inflammation of peripheral nerves. You get kind of somewhat paralyzed and you get a lot of pain. And these are two cases where the individuals had these symptoms and they were shown to have Zika virus infection, probably by PCR, and negative for lots of other viruses. And they had virus in the urine over 15 days after the onset of symptoms.
Is this the first time that I was amazed that I had not come across this term before, of viruria. I mean, it's obvious what it is.
That was new to me.
Yeah,
you should have both come across it a long time ago.
Yeah, it's old.
Somehow I had not.
Yeah, it's a cool term, isn't it?
It is.
Mm-hmm.
I like it.
It's related to viremia, for those of you who want the linguistic breakdown, and it's instead of the eem from heme, it's the from urine.
Right, which is, I mean, that's standard construction for these terms. Anything that's urea is in urine. Anything that's emia is in blood.
Yep.
Right.
Yep.
But
so what I found interesting here, two things. First, the reported quote-unquote sexual transmission may be related to virus in the urine, right? Because I'm not going to go into it, but you can imagine how that would happen, right?
Yeah.
Mm-hmm.
Yeah.
Good. And secondly, West Nile virus has been shown to be in the urine as well, another flavivirus. And I'm wondering if Zika is actually replicating in the kidney or if it's passing through, right, from the blood. I think that would be interesting. I don't know how you'd find out because no one's going to let you biopsy your kidney.
need a good animal model.
But it would be interesting to know how many more of these, and how many infections does this occur? Is it only in symptomatic infections or all? So interesting. But I think the most important thing is in terms of transmission, because not only is the world covered with a thin layer of fecal material, but there's urine everywhere. And
there's also skin everywhere.
There's skin everywhere. I told my class that last week, and they were not very happy about that. All that white stuff you see, what you think is dust, is your skin.
I just covered an urban microbiology meeting at the New York Academy of Sciences that was held last summer, and one of the things that they discussed was how the whole subway system in New York is covered with skin.
Yep,
and it has the skin microbiome all over everything. But yes, there's also feces and urine everywhere.
Yeah, so urine contaminates things, and when you go into a public bathroom, and you flush the toilet, there's always an aerosol. So urine could be one way. And
so, are you saying Zika virus is an aerosol infection now?
I don't see why it couldn't be in some cases, because your nose. You remember the soft palate? There's a mucosal layer that gets infected with influenza virus.
Has Osterholm already hyped this?
No, but he did something else that was objective.
Give him a moment.
On day 0, he said he was convinced. So anyway, and then there's a second paper from the Journal of Clinical Virology, which also shows prolonged detection of Zika virus RNA in urine samples during the epidemic in Brazil. So in this case, the RNA was detectable in urine 4 days after onset of symptoms and observed until 14 days after onset.
And these others they observed RNA in the urine 15 days after onset of symptoms. So this is beyond what we typically think of as the time when the acute infection is over and resolved. So we have a couple of different things here. We got virus in the urine for at least 2 weeks, and we got virus in semen for up to 8 weeks, at least in one case. Okay.
Well, at least by PCR.
PCR anyway. So that's
by PCR.
That's the issue. In these urines. They need to see if there's infectious virus there at some point, right? Because no one has, I think, yet found infectious virus in urine. And if there's not, then it's not a transmission risk. So we need to know that. So please take some of this, filter it, and put it on some Vero cells. Not that hard.
So I need to educate myself here because I'm confused on an issue. And it's basically the difference between a peripheral nervous system infection and a central nervous system infection. So Guillain-Barre, is that, and by central nervous system, we're talking about brain and spinal cord. Is that correct?
Yeah, that's right. That's right.
And peripheral nervous system is anything that branches out from there.
Yep.
Right. Are there infections? Yeah, sure. There are infections that are there compartmentalized to one or another of those compartments?
Well, herpes viruses go into the peripheral nervous system, right?
Right.
That's one comes to mind.
And they stay there except under unusual circumstances where they can invade the CNS and cause encephalitis and stuff. Guillain-Barre is a CNS infection. Is that correct?
No, Guillain-Barre is a peripheral. It's a inflammation of peripheral nerves. So we don't even know if there is replication. And remember, not just viruses cause it, but bacteria infection as well.
Right, because we're talking about basically sort of inflammation. Call it that.
Yeah,
right. Guillain-Barre is it's not specifically an infection; it's a syndrome.
Yeah.
Okay.
Right.
So it's diagnosed based on
the word autoimmunity comes to mind.
Right.
Yeah.
Right. Yes, that's what I was thinking of. Okay.
But it's associated typically with a variety of microbial infections. Yeah.
All right. So even though it presents as neurologic, it does not necessarily mean that whatever the pathogen is infecting nervous tissue.
Right.
Okay, good.
It could mean the pathogen triggered an autoimmune response.
Right. Okay, good. Because we do have, stuff coming up with Zika about interactions with CNS or peripheral nervous system.
Yeah.
Right. So I want to keep this in mind as we go through this stuff to make these distinctions.
Yep.
Okay.
Before we leave the urine story, I just want to point out that the authors mentioned that urine samples could be considered as an important alternative to serum or plasma for detecting Zika virus RNA because of the longer period, higher RNA levels, and that it's less invasive.
Easily accessible.
To collect the sample.
The next Zika report. So remember last Friday's TWiV, we had just gotten the New England Journal paper.
Right.
Actually, it came out after we...
Right. We only had the abstract.
Right.
So this is Zika virus infection in pregnant women in Rio, a preliminary report. Alan, is there any other... It's now out, so is there anything else you wanted to point out about this?
Right. So right after the podcast ended, I checked again, and finally, the embargo was up, and they released the whole thing. But it's, it's a frustrating report. It's great that they're doing this, and I'm glad they're following it. There's not a whole lot that I think we can conclude on the basis of this report. I understand why the NEJM went ahead and ran it, but it is very much a preliminary report. It's properly titled.
One of the things that we were frustrated by in the abstract was that it said 8 of the 42 women in whom the fetal ultrasonography was performed have delivered their babies, and the ultrasonographic findings have been confirmed. And we wanted to know,
confirmed good, confirmed bad, which outcomes did you confirm? Yes.
Even with the full data, it's still a little hard to make out, but it seems like, some of the ones where they saw abnormalities in ultrasound, that played out in the births as well.
Yes. This is certainly not a slam dunk for Zika virus causing microcephaly or anything else, but it does show the right kind of exploration to find that, the right kind of epidemiological analysis. There's just not enough here.
Right, and because at the time of this writing, only 8 had delivered their babies. When we get the other 34, we may have more data.
Right, and I gather from the way this paper is written that this is not, we're done now.
Yeah,
this is, we are continuing to sample and follow this entire cohort and enroll more patients, so they're going to continue monitoring this and presumably releasing reports like this periodically. So that part's great.
Right, and as you remember, it seemed pretty clear that, of the 42 Zika virus positive women, they detected 12 of them having some kind of abnormality by ultrasound and 0 in the 16 negative women.
That's right.
And that, seems like, it's kind of to me like when there's a clinical trial and there's a drug result and it's looking like it's all going one way, that you sort of at least report some of it, or you maybe change what you're doing or something. And so I think that they would have been faulted had they not presented some of this data, maybe.
Yes. Yeah, as I say, I think it was appropriate. It's certainly appropriate for them to be doing this, and I guess it's appropriate to issue the reports as they come.
And they do title the paper preliminary.
Preliminary report, yes.
I have to say
I don't see why this needed an embargo, though.
When you read the abstract last week, I kind of held my tongue until I saw the paper. But having seen it now, I have to say that 29% having abnormalities of one sort or another, to me, is a big number.
It's kind of high, I think. Yeah.
Right.
Yeah. But it's not even that high in rubella. Yeah.
It's chilling.
Mm-hmm. So
I also note that this group apparently has a long-standing function of this type in monitoring dengue. And so they've got everything in place, and they've morphed it over into Zika. So this is, as you say, not a one-shot deal. We can expect these guys to keep going on this for a period of time.
Right, and in fact, they find a lot of the women in this study are double positive for dengue and Zika.
Yes,
which is not, of course, is not the least bit surprising.
But they don't stratify the outcomes based on that at all. You know,
right?
So I don't know which of the women in the Zika positive and dengue positive had defective fetuses and et cetera. They don't do that at all.
Right, but when you're working with when N is just over 3 dozen,
yeah, of course,
you start stratifying, and pretty soon you've got groups of 1.
So I was sent on Twitter a article by a blogger. His blog is called Epi Ramble. His name is Andrew Lover, and it's an interesting analysis where he says he understands it's a preliminary report, and they're probably doing this under very difficult conditions. But the title should really be "Risk of Any Adverse Fetal Event in Pregnant Women Who Present with Rash and Agree to Follow Up," because that's what he says. It's not clear what the measured outcome is here, but any measured malformation appears to be the closest. This is a major issue. Without a well-defined outcome to measure, any abnormality becomes a study event. Clearly, some are major. However, one of the reported cases has megacisterna magna, which is of very limited impact, he says. He also says that the study wasn't blinded. He doesn't know if the people doing the sonography were blinded as to the mother's Zika status, which of course is powered. And then he says it's not well-powered, and a few other things. So he says, I think it's clear from the very large number of adverse events that something serious is occurring in this cohort, but I don't think these results make a watertight case for Zika in isolation.
Right.
I suspect that there are other things going on here, but I'm struck that the Zika negative women had 0 abnormalities, right? So that's what I stick with. All right, so then last week we also had 2 reports of Zika virus in the central nervous system. And the one report, this is from, this is a very interesting one, yes. An 81-year-old man who lived in France. He had just returned from a 4-week cruise, New Caledonia, Vanuatu, Solomon Islands, New Zealand. And the moral here is stay home, of course. And then a day later, he developed a fever and hemiplegia of one side, weakness of one side, the left side, paresis of the right upper limb, and he had to be intubated because he wasn't breathing well. And then he developed a rash. And
he went into a coma.
Went into a coma also, yes. The Glasgow Coma Scale, score of 6 on a scale of 3 to 15. And I just want to note that I'm usually around 15 on a daily basis. MRI he has showed he had meningoencephalitis, which is a inflammation of the meninges, the membranes surrounding the CNS, and the brain proper, the cortex. And they have nice MRIs there. CSF lumbar puncture, cerebrospinal fluid contained Zika virus by PCR, Zika viral RNAs, I should say, by PCR. And then they took some of the CSF and put it onto Vero cells and outgrew Zika virus.
Right. So now you got virus.
Very nice, guys and girls. Very nice. I like that. So this is quite
from cerebrospinal fluid. So they did a spinal tap.
Yeah.
Right? And grew virus out of that.
Yeah.
Mm-hmm.
And so we're talking central nervous system, and we got virus in it.
Yeah.
Yes.
So that's the first one I know of the CNS, but we'll see if there's more. Now, this is an 81-year-old man, so it may be a thing you see in older people with reduced immune system, but we'll see. All right, now, the next one kind of addresses this because this is a young girl. Ooh, I didn't put the link in, Kathy?
No, not in your blog post.
In the blog post.
By the way, did you note that this guy recovered fully?
He recovered fully, right. Thank you, Kathy.
Unbelievable.
Yeah, it's good that he did. And he's probably happy. You don't want to come back from a vacation and die, right?
No.
I'd prefer not to. I'd rather do that than die on the vacation, I think. Well, it doesn't make any difference.
Yeah, you're being morbid now.
I'm sorry.
This is published in The Lancet, and it's acute myelitis due to Zika virus infection. This is a 15-year-old girl admitted to hospital in Guadeloupe, and she had arm pain, frontal headaches, and other kind of CNS indications. Lower back pain, weakness in her left arm, no coma. Okay? No fever. And then she developed an inability to urinate properly, so they catheterized her. And they thought this had something to do with nerves. They ruled out other reasons for urine retention. And they found Zika virus by PCR in her serum, urine, and cerebrospinal fluid. And they looked for other viruses, and they were all negative, viruses and bacteria. And they show a nice MRI of her spinal cord showing myelitis, an inflammation of the myelin sheath. So another apparent invasion of Zika into the spinal cord, this time in a 15-year-old.
Right.
And they tested for varicella zoster and herpes simplex viruses, Legionella, Mycoplasma pneumoniae, and that was all negative. And she had no signs of acute infection with cytomegalovirus, Epstein-Barr virus, chikungunya or dengue, syphilis, Lyme disease, HIV or HTLV. And is this the one that... Oh, no, that's different. I was thinking this was the one that they had her on some antivirals, but that's a different story. Never mind.
So this is, to me, this is not too surprising because other flavies do enter the CNS. West Nile virus in a small fraction of people, the virus gets in the CNS and their neurological symptoms. Japanese encephalitis virus, of course, it's in the name. Dengue. Also tick-borne encephalitis virus, a really serious CNS infection. Flavivirus also. So, these other viruses, West Nile in particular, causes a wide range of things from myelitis to acute flaccid paralysis. So if we get more cases of Zika, we will know the whole range of what's going on here.
Right.
Quite interesting. Okay, and the next up on our Zika report, I found a morbidity and mortality weekly report from the CDC. Transmission of Zika virus through sexual contact with travelers to areas of ongoing transmission. These are 3-case reports of individuals who, got infected and then had sex, and the recipient, condomless sex in all 3 cases, and the recipient also got infected.
And the recipients are always females.
Yeah. So, and these are like the previous cases. CDC says this is probable sexual transmission, but it seems to me if viruses in the saliva, urine, and even in the rash, maybe, I think one of these cases, the person still had a rash when he had sex. Really smart. The rash could be contaminating the partner. So while it looks like sex, I don't know why all right, I know why they're not ruling it out. Other things at this point, sorry, I said that wrong. I understand why they're making a big deal of the sexual transmission, because they don't want to find out in 6 months that it is sexually transmitted, and then a lot of people will have transmitted it to each other, transmissible. But I think we don't know yet. Even if it is, I think it's probably rare.
And it's, I think it's important to note that in all of the documented cases of supposed sexual transmission, they've happened either while the person was symptomatic or very shortly after they'd recovered. So this is not, we don't have any reports. It's not that it's impossible, but there are these, with Ebola we saw this, with Zika we're starting to see this, where people can detect by PCR virus in the semen for days and weeks and maybe months after the patient is recovered. We don't see sexually transmitted cases occurring 3 months after somebody recovered from Zika virus.
Yeah. What I'd like to see is infectious virus in semen, right?
Yes.
And that was found once in a French Polynesian patient, but he didn't transmit it to anyone. They just found infectious virus there. So, okay, it could be there. But in these cases of supposed sexual transmission, I think you have to find infectious virus in semen and then the same virus in the recipient. And if you find that a number of times, then I'd be convinced. That's hard to do, obviously, right?
Yes.
Yeah, and it would be most convincing as if the transmission occurred at some late period after the infection. So when, the most convincing would be when you've got some signal of virus, in particular infectious virus, in the semen and nothing in the serum or elsewhere.
Right.
Okay?
Okay, and now also from Twitter, someone sent me, actually it was on my blog, someone emailed 2 papers from the Pakistan Journal of Zoology, which I'm admitting I don't read regularly. Teratogenic effect of sublethal doses of deltamethrin and beta-cyfluorothin, which are pyrethroid insecticides of the nature of the sort that are being put in the water in the northeast of Brazil. Correct, Alan?
These are different.
They're different?
Is so the ones. It's a different class of insecticides that were being used as larvicides. Those are specifically for killing mosquito larvae, and especially in places that people might be drinking the water.
Okay.
And that's that. We talked about those already. Pyrethroids are the class of insecticide. They're probably the most commonly used insecticides in the world. If you go to the hardware store and you go to buy bug spray, you'll see a big shelf of all kinds of options, and they are almost all different variations on pyrethroids. So this is a very, very commonly used class of insecticides. They're used for indoor spraying. They're occasionally used on crops, although I think a lot of agriculture has moved on to other things. This, by the way, the pyrethroids were the second major class of insecticides my grandfather worked on, so I've actually spent some time reading about these.
Good.
The 2 papers, these are from the same group in Pakistan, and they're really, between the 2 papers, I think they have about 1.5 experiments. There's, and it's really hard to make much of what's going on here. So they fed mice pyrethroids, permethrin, 2 different permethrin derivatives, and they fed them in one of them. They actually bothered to do an LD50 and plot the data, and they came out with an LD50 for this for deltamethrin that is an order of magnitude lower dose than the LD50 that's been found by everybody else who's published on this.
Mm-hmm.
So I don't know. That's weird, obviously, and it's possible that their prep of deltamethrin was contaminated with something. Anyway, and it's clear from the way the papers are written that they're kind of on a mission to say that the pesticides are causing birth defects.
Yeah.
I mean, they're grasping at all kinds of epidemiological straws that are studies that are almost equally thin as these 2. To say that insecticides are bad. Okay. And it's just, yeah.
The ones that are used in Brazil are pyroproxifens, right?
Yes, the pyroproxifen is the larvicide that's used in drinking water, and that one I was not familiar with until it came up in this context.
And that's different from these, chemically different.
That's different
right?
from these,
right,
yes.
Thank you.
That's and the permethrin gets sprayed all over the place. So these folks dosed mice with permethrin, and they found they picked a dose that even if you take their own LD50 data, the dose was actually kind of high. So just because it's sublethal, I guess that was their standard, and they gave sublethal doses of this, whatever it is, and they saw various types of fetal malformations and defects in the mice, and that's it. That's both papers, and I don't think either one of them amounts to anything useful.
All right, that's great. Thank you very much.
Sure.
All right, the last thing is something that came in just an hour ago. A company called Visual Science sent me a link to some Zika virus illustrations that they've done. These are computer-generated illustrations and animations, basically, and they're beautiful. You can
they're really nice.
Go to the illustration, and there's some circles you can click on that zooms in.
Neat.
And these are based. So we know some flavivirus structures. Obviously, we don't know the structure of Zika virus yet, but these are based on those flavivirus structures. So they're quite pretty. and there's a movie as well. So check that out. And we've had this as a pick from other people before. This company, Visual Science Viral Park, is their project, and they have other viruses too. And they also have some plastic models, which are gorgeous, and I would like to have, but they don't sell them. I guess you could print them out, but they're pretty cool. There's a plastic HIV and DNA, etc. So thank you.
Oh, they've got an adenovirus, Kathy.
They have an adenovirus.
Yeah.
It's very nice. Okay.
That would look good on top of a Christmas tree.
It would. All right, so that's our Zika report. Could actually break this out and release a separate Zika report every week, right? Dun, 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 dun. Okay, so now a snippet. For this, I was looking for a plant virus paper, and of course I had an email from our friend Steen, who sent in a few weeks ago. Dear hosts and microbes, please consider discussing a paper that uses recent improvements in electron cryo microscopy. I see that you have discussed cryoEM structures before, TWiV 101, but it sounds like there have been substantial improvements since then. He gives us links to 4 reviews, and he suggested a structure of cowpea mosaic virus. This open access paper came out a few months ago. A press release with a link to a video just appeared. The paper has a nice overview of the genome and structure of the virus particle and a cartoon of how assembly might work. The authors are interested in using cowpea mosaic virus. Virions for drug delivery and infectious CPMV clones are an amazing tool for driving protein production in plants, including antibodies and antigens for vaccines, which can accumulate to 20% total extractable protein. Wow, that's some impressive hijacking of translation. P.S. I listened to TWiV to hear the latest news on reverse genetic interrogation of mutant protein expression during infection. And, this was part of the textbook contest, and I don't think I included that. It's really good.
That is good.
Yeah.
It might deserve a book, don't you think?
Yeah, it pretty much should push all of your buttons and all of Jane's buttons.
Does. I don't know, Steen, do you need a book? Maybe you don't. Yeah, maybe you don't need a book. Anyway, so this is a cool paper, and it involves cryo-electron microscopy. And to review that, basically, you take a virus, anything you want to solve the structure of, but here it's a virus particle. And instead of doing X-ray crystallography, where you need to make crystals, which is really hard, and then they have to diffract X-ray beams, you just take the purified virus particles and you freeze them very quickly in an aqueous solution, and the freezing gives them enough contrast so that when you look at them in the electron microscope, which has to be cold, of course, you can see the particle in some structure. So you take lots of pictures, hundreds and hundreds of thousands of pictures, and the assumption is every particle is in a slightly different orientation, and then you do math and you use the different orientations to generate a three-dimensional image, sort of like a CAT scan, I guess, right? And
it works if you have symmetrical viruses.
That's right. And you can also do a version of this where instead of having to take so many pictures, you actually tilt the image on the stage, and that's cryo-electron tomography, and you can get a lot of different angles that way as well. And this has improved so much. I remember years ago I collaborated and we did with Jim Hogle and Alastair Stephen, we did polio plus its receptor by cryoEM at like 9 angstroms. Now you can get 3 angstroms and below. Largely, I understand because the computational approaches have been really refined and they're really better.
There's a little video that goes along with this link that I looked at, and the implication is that the instrumentation is significantly improved.
Yeah, probably that too. Yeah.
Yeah,
they pay a lot of money for these big cryoEM. You have one over at Michigan, Kathy? You must, right?
Mm-hmm. Mm-hmm.
Is it new or old?
I don't know. I just know that we have at least one cryoEM expert.
Yeah.
And so I don't know the instrumentation.
So here they do cowpea mosaic virus. This is actually kind of close to picornaviruses. It's in an order being, as far as classification goes, a collection of families called the Picornavirales. So orders end with the alleles. And that includes picornas and these cowpea mosaic viruses. And so there are over 100 structures of these viruses, but they're missing some things like, in this case, the RNA. They'd like to know how it's packaged because they want to use this virus, as Steen said, as a vector. and they don't know how to package. They don't know anything about packaging. In fact, in all the picornaviruses, we've never identified a packaging signal, something in the genome that gets it specifically into the capsid. And this, of course, has been done for adenoviruses, SV40, retroviruses, and other viruses. And you can use that to put foreign genes in the capsid, right? But you can't do it with picornas. So they're hoping if they get a better structure, that'll be something that comes out of it. And as you heard from Steen, you can really make a lot of protein in cells infected with these viruses. Now, they're very cool. They are positive sense icosahedral particles, single-stranded RNAs. They're 2 RNA segments. They are separately encapsulated. They're about 30 nanometer particles, and you have to be infected with both to get a productive infection. Now, that if you don't scratch your head after that, why would this happen that you need to you package them separately and you need both? Is that's nature's secret? Who knows? But it works apparently, right?
And these aren't the only viruses that do this.
Yes, of course.
Right. But I think this is. I wonder if this is more common in plant viruses than other viruses, because I know of I can't name you chapter and verse, but I know of at least one other plant virus that does this 2-particle thing. But
yeah.
I don't know.
The Gemini variety. Yeah, they're DNA viruses.
It may be because of the way they're infected, insects. The
way they're delivered,
right?
Yeah.
You get a lot of particles at once, so that supports this. Maybe. That's a good question. Yeah.
Well, actually, now the Gemini variety, the viral zone is showing a sort of a connected capsid, double icosahedron.
Right, but I think there are others that have a 2-particle thing.
Yeah.
Right, and I could see if the virus has evolved a life cycle where it's producing huge titers, there wouldn't be any disadvantage to requiring 2 particles to get in, because it's going to happen. You're effectively doing a high MOI infection every round, and there could be advantages to making the capsid smaller.
Maybe.
So yeah, that could tilt it in favor. But yeah, it's a pretty bizarre adaptation.
So the question we have, which we can't answer, are there any non-plant viruses with bipartite genomes, right?
That are delivered separately. Yeah, offhand I don't know, but probably if you parse through all of viral zone, you can answer the question.
The only example of a 2-particle hit thing in animal virology that I can think of is defective tuber retroviruses. Okay, but I think of that as sort of almost a laboratory artifact. I don't know if that's accurate or not. That's the way I think of it.
I mean, of course, there are helper situations like hepatitis delta virus, which is packaged, but it's not one virus with 2 different genomes. Yeah, I mean that delta requires hepatitis B infection, but it's a different virus. So
although maybe that's the way you get from. Maybe that's the way you get to an evolutionary endpoint, like with Cowpea mosaic.
It could be.
Okay, listeners, especially you plant virus dudes, help us
out. Are there any out there?
Yeah, I don't know. We don't talk about plant viruses enough. Maybe we've lost them. Wait, come back.
All right, so Cowpea mosaic virus, the capsid is an icosahedron. There are two capsids, of course, and the capsids are made of
so you have to get 2 pods in a P.
You have to get 2 pods. There you go. 2 pods in a P. It's a good title. The capsid is made of 60 copies of a large and a small coat protein that are processed from a precursor. And there are turrets at the 5-fold axes of the particle. There's a part of this structure that they don't have, and that's what they're trying to get here. It's the C-terminal part of one of the capsid proteins that's been implicated in RNA packaging. And this thing is processed away in the mature particles, so in all the previous structures, they don't have it. So they're trying to get it here. And they tried to do it in a different way to get the structure here. So they want to somehow prevent this C-terminal cleavage, so they make empty virus-like particles where the cleavage is slower. And so they figured maybe we could see some of this C-terminal protein present in this preparation. So they took pictures of 62,500 particles. Can you imagine? It's a lot, but in terms of viruses, that's nothing. But they took all these pictures and then they used their computational methods to end up with a very nice structure of, 3 and 3.8 angstroms for something for the empty, and then also they did the full capsid as well. And in fact, they do see this C-terminal region for the first time ever. I can imagine how excited they were. And the C-terminal region is in the interface between the subunits in the turret. I have a lovely picture showing that. And they can tell from that what amino acids this little C-terminal segment is interacting with. And so they can change it and see what happens. And so when you change the sequence, the C-terminal sequence, and the surrounding sequences, you can mess with packaging of the RNA. So this is clearly involved in packaging.
Yeah, they show a really nice image of the mutant virus, and there's no nucleic acid in it. It's just
it's pretty
amazing.
cool. And you can make, and of course, they can see the empty particles because they make so many of them, and you can see that there's no RNA in it. It's very cool. Then they also, in some of these, in their structure with the RNA in it, they actually see RNA density, which has been found for other RNA viruses before. It's hard to see it because typically the RNA is not ordered, so it doesn't show up like the capsid proteins do, but they see concentric shells of density, which is the RNA, and you can see them very nicely in this paper. And it looks like it's duplex, double-stranded nucleic acid, and it seems to be extensively base paired, and so they think that may have something to do with packaging. And the strongest RNA density is right below the twofold axis of symmetry, and so they mess with the amino acids under there, they change them, and in fact that interferes with RNA packaging. So by having the RNA density, they're able to identify regions where they think the RNA is interacting with the capsid, and they can say these amino acids in the capsid are important for packaging. Of course, we don't know what parts of the RNA are important because you can't get enough structure from this, enough precise structure to do that. But it's a start, I suppose.
Right. They back on those shells, they say the RNA is these concentric shells, but this density is an icosahedrally averaged picture of an asymmetric RNA molecule, so they can't interpret it. But there was something else I was going to say about that. Oh, that because of the thickness of the shells, it suggests that there's extensive base pairing during encapsulation, that the RNA is double-stranded for a large part of it, I guess, is the way I interpreted that.
I wonder if you could do mutations that would be mild enough that you could select for virus that would get around those capsid mutations and maybe find the RNA residues that way.
Yeah, that's not a bad idea. Yeah, so even the ones they have, maybe if you push hard enough, you could get some encapsulated. There may be a small amount of encapsulated virus in there. Yeah, that's a good experiment. They're probably doing it. So they have a model for assembly, which is very much like the picornavirus model. The capsid protein, the two capsid proteins are a monomer, basically. They form a penton, an assembly of 5, and then the pentons can go one of two ways. They can assemble with each other to form an empty particle, or they can combine with the RNA via this sequence at the twofold axis that they've identified, and then get incorporated into the genome. And it's exactly the same model that we suspect polio does, because we find empty capsids as well as RNA-filled capsids in infected cells. So it's pretty cool. And I have something in my heart for structure because very early in my years working on polio, Jim Hogle entered the field and determined the X-ray crystal structure of polio. And I got that early on and collaborated with him for many years, and so it's kind of driven our work. It's cool stuff.
Was that like the first whole virus structure?
So the first RNA virus was a plant virus that Steve Harrison did in 1976. Tomato bushy stunt virus.
Okay.
And then, of course, Jim Hogle worked with Steve Harrison. Jim did polio in 1985. At the same time, Michael Rossman did rhinovirus.
Okay.
I remember as a postdoc, I was at MIT in 80, And we had just done the sequence, and he called me up and he said, could you give me the sequence of polio? Because we figure we're going to solve the structure in about 10 years, and we'd like to have the sequence. And it turned out he did it in 5 years or 4 years. But the other thing was, I said, okay, how do I send it to you? And he said, just give it to me on a computer tape.
Computer tape.
And I had this like 12-inch diameter reel of tape, right? And I brought it to the computer center, and they copied it onto it and mailed it to him.
God, amazing.
Can you imagine?
Well, I remember, I'm sure you must still have this somewhere, but it was posted outside the lab for a while. It was a poster that you'd presented at a meeting, and you had the sequence typed out and pasted onto the poster.
Yeah, I made that at MIT. We had to print out the sequence, and I cut it up and pasted it on a big board because there's no way to make any kind of graphics, right? And then we photographed it, and that was published in the paper. And yeah, I had it around. It's gone now.
Oh.
It's too bad. I had hundreds and hundreds of sequencing gels, X-rays, and I threw them all out. And now I'd like to have one just for memory, but I threw them all out.
Oh, I had boxes of those myself just for my PhD work.
You threw them out?
I think I left them somewhere, but hopefully somebody threw them out.
Yeah, they're gone.
So I may have told this story before, but when I went to MIT to do a sabbatical in Phil Sharp's lab, I took over Claire Moore's bench. And she was into polyadenylation. She did in vitro polyadenylation assays, which were run on sequencing gels. So these things look like sequencing gels. And the first event that I went to was a going away party for her at Phil's house. And as a going away present, somebody had raided a bunch of her old films and made this remarkable lampshade.
Oh, how nice.
Oh, nice.
Out of all these old, essentially sequencing films. And it was beautiful.
That's great. That's
neat.
Great. I don't think I said the name of this paper, Mechanisms of Assembly and Genome Packaging in an RNA Virus Revealed by High-Resolution Cryo-EM. The authors are Hesketh, Mesheriakova, Dent, Saxena, Thompson, Cockburn, Lomonosov, and Ranson. They are from the Norwich Research Park in the UK and Indiana University. Thank you, Steen.
So very nice. Plant virus. There you go.
Yes,
very
cool.
Nice stuff. Our paper today is a letter in Nature, and it's called, many people sent this in because it's pretty neat, MIMIVIRE, a new name. MIMIVIRE is a defense system in mimivirus that confers resistance to virophage. The authors are Levasseur, Beckles, Chabrière, Pontarotti, Lascola, and Raoul. Sorry for the pronunciation. They are from the Aix-Marseille Université in France, Marseille. Listen to this wonderful name. Unité de recherche sur les maladies infectieuses et tropicales émergentes. I know that's not the way you say it, but emerging tropical infectious diseases. So mimiviruses, remember the big DNA viruses discovered now over 10 years ago. And then subsequently they found virophages that will only replicate in amoeba, one of the hosts of these, or the known hosts are amoeba polyphaga or acanthamoeba polyphagia. The virophages only infect cells infected with mimiviruses, and they actually reduce the yield of the mimivirus. So they're called virophages, and that name has stuck.
So it's kind of a virus that infects a virus. Yeah.
Right. So
Kind of.
Kind of. Right. I said that so that Vincent wouldn't go off, but
But we did.
yes.
Are you saying I go off sometimes?
only occasionally.
Oh, I don't want to do that. That's annoying. I'm sorry. Yeah, they infect virus-infected cells.
Right.
They recently there lots of these virophages have been found. In fact, if you just query or interrogate, is that the word?
Yeah.
If you interrogate the global ocean explorer data sets where what's his name sequenced the ocean, you can find lots of virophage sequences. So this group found a new one called Zamillon, and they said, this is the interesting thing, there are three lineages of mimiviruses, and Zamion, the virophage, only infects strains of the B and C lineage, but not A lineage strains. And so there's some specificity. And so they're thinking, maybe this has to do with the defense system of the MIMIVIRE. Hello, Dickson.
Now, the name.
Hello, everybody.
I'm sure this is probably a French pun on something, right?
Zamion?
It's because it's not any kind of a strain number or anything like that.
I mean, is it a place?
I don't know.
I
Googling it
and
only
I got
gets
nothing.
this.
Really?
Yeah.
Qu'est-ce que c'est, Zamion?
Je ne pas.
You don't know how to speak French, do you?
This is very
How are you, Dickson? Are you well?
Thank you. I'm well. So I'm a little bit late. The class ran a little long.
We were
today.
talking about the MIMIVIRE.
Yeah, no, I read the paper. It's very exciting, actually.
Hi, Dickson.
Hello there, Richard.
Hey, Dickson.
Hey, Alan.
So
Hey,
what
Kathy.
they're thinking is that, and it probably didn't happen in this order, but that's the way they write it, right? They think maybe it's a CRISPR-like system in the MIMIVIRE that's blocking the virophage infection. And just to remind everyone, this CRISPR-Cas is an old intrinsic defense system that's been found in a lot of archaea and about half of all bacteria, where they retain a memory of infections by keeping pieces of the infecting virus genome and putting them in their genome. And then later, when they get reinfected, they can target that genome with RNA-based nucleases and protect themselves. So it's an old defense system. So they're thinking maybe there are CRISPRs here. So they took 45 MIMIVIRE sequences and they screened them for virophage DNA. So this is computational screening. They find a 28 nucleotide stretch identical to Zamion in the lineage A MIMIVIRuses. How much better can it get?
Not much.
Not.
So remember,
Exactly.
the lineage A is not infected with this Zamion.
It's resistant to this, right?
And they
found this same stretch in one of the lineage C viruses. Is that correct?
Correct.
Mm-hmm.
Okay, but this is before the plot's still a little thin here. It'll get thicker.
All right, so this is a perfect match for the Zamion genome, right? And then they find a 15 nucleotide sequence. By the
way, they have a positive control, which I really like.
Yes.
Which is the original Sputnik virophage that infects all these things, and that's handy. I like that.
Mm-hmm.
It's absent in Sputnik, this sequence, right? That's what you're saying, right?
Right, and Sputnik infects
all of them, right?
Right, is not excluded by any of these MIMIVIRuses.
Yeah.
Right.
So then they find that 15 of these bases are repeated 4 times in the A group genomes and not in the B and C genomes. So that one C example that you mentioned, Rich, it's not repeated there.
Right.
That's the wrinkle you were referring to.
That's
right?
that's the thickener.
The thickener.
So when I read this, I wondered whether or not the defense system is geared towards the most frequent invaders, and whether these other B and C strains are less prevalent, and therefore the development of a defense system would be newer.
Well, it's all about ecosystems, right, Dickson?
Yeah.
We don't know where these are.
Well, it's an intracellular.
No, and I mean geographically, if they're separated, they would never encounter each other. Right?
Well, that's absolutely right.
Good point. It could even have to do with the actual geographic distribution of the viruses.
But this virus lives in the cytoplasm of an amoeba.
Well, that's the experimental model.
So then you wonder how did the invader viruses get to the MIMIVIRE through this amoeba? Maybe
this is. I asked the authors years ago, is amoeba the real host? And they said, well, it's the only thing we found that can be infected, So who knows if it's really the host out there?
The only thing you can work with in the lab that you can infect with this, but of course there's this huge unculturable world out there.
Now here's what gets really cool. They look at the sequence around these 15 nucleotide repeats. So again, this is the Zamion sequence repeated in the MIMIVIRE genome.
Yeah.
They find a putative phage-type endonuclease, which is related to a Cas nuclease, which is involved in CRISPR, right? They find a putative helicase that's now helicase would be needed to denature the incoming virus so that it can be chopped up by the CRISPR system. and this is related to a Cas3, which is in the CRISPR-Cas system. And then they find an RNase 3, which is related again to a CRISPR-Cas gene. And the RNase, of course, chops up the RNA transcripts that are made from the repeated CRISPR loci to make the targeting RNAs. So they got a bloody, and the arrangement is very similar as in the Cas, right? There's some differences that they go into, but it's just amazing. It's like it's got its own CRISPR-like defense system.
Right.
Yeah.
So in fact, there are these enzymes, and then the DNA is repeated and spaced out. It's a little different the way it's spaced, but pretty darn cool. And they don't find this in any of the other megavirale families outside of MIMIVIRE, which is quite interesting.
They probably just haven't found the right phage for it yet.
Yeah.
Yeah, good point.
They're probably not.
There's only a couple of these.
Because everything. This is just reminding us that everything gets viruses and everything develops immunity. It's if there's a virus infecting it, then there's some kind of response from the. And yes, you could argue that the virus is not infecting the MIMIVIRE, but it's these two are interacting in a way that is very much like an infection.
I guess they're going through a phage, right?
Yes, they're going through a phage.
Kat, you didn't like that?
Right.
I have two bits of lateral thinking. One is that when I went to the second page of Google, I found a different kind of a hit where there's a book called The City of Saints and Madmen, fantasy short stories by American writer Jeff Vandermeer, set in the fictional metropolis of Ambergris. And anyway, there's a text bit about the ruins of Alfar form, along with Zamalon or Zamillon, the only recorded instances of a particular architectural style reminiscent of gray cap buildings. So they may have gotten it from this fantasy science thing. So that's the first one.
Can I just ask you a question? Do you routinely go to page 2 of Google? Because I never do.
Oh, no.
That is a dark zone.
Right.
Dark zone.
But I just thought, there must be something more. So I went to page 2, and that's what I found. But I found a lot more instances of, MIMIVIRE,
right.
Zamion. So, yeah. The second bit was back when we were talking about MIMIVIRUses being very large, and they're so big that you can see them with the light microscope, except that these authors talked about the MIMIVIRUses being visible with photonic microscopy.
Yes.
And so I thought, I think that's probably, a translation from French of how they talk about light microscopy. So in Google Translate, I found that they either call it optical microscopy or they call it photonic microscopy, what we call light microscopy.
It's cute. Yeah, do you think the Nature editor left it in because they thought it was cute?
Yeah, I don't know.
I hope
It's
so.
Star Trek-y. It's very Star
Well,
Trek-y.
actually, about 6 lines before that, they say in this former example, and I'm sure they mean in the latter example. So I'm not sure how well the Nature editors read.
Oops.
All right, so the next thing they did is to ask, are these CRISPR-like sequences functional? So they silence them by using a short interfering RNA. So you could put RNAs into these infected cells and the amoeba, and then ask, is silencing of any particular one of these genes associated with an increase in virophage replication, right? You're taking away something's crucial. And they silenced 27 different genes and assay virophage replication, and what they find is that you get more virophage DNA after silencing the endonuclease, the helicase, and the helicase. That's the only one, right? The endonuclease and the helicase. Those are the two.
Right. The ribonuclease doesn't have an effect.
RNase 3, didn't have an effect.
And the biggest effect is when you knock out the gene that actually contains the 15 base pair repeats.
That's right. It's right, exactly. The repeats themselves, they also silence. And so these are functioning in some kind of defense. They also produce the nuclease and helicases by producing the proteins individually, and they assay their activities enzymatically. This is quite nice that they did this, right?
Yes.
Actually produce the proteins. And they show that the the nuclease actually cleaves DNA, and the helicase can unwind DNA, so they have the right biochemical activities. So that is pretty cool. Now, the last question, where did these come from? So,
exactly.
I hate when I say so every time. I do it all the time, and I can't stop.
I don't care.
I need therapy. I hear myself all the time, and it's just terrible. All right, we're going to try and systematically avoid saying it.
I wonder if. Well, okay, go ahead.
No, you probably.
No. I was about to let go of a secret, so I'm not going to do that.
But don't you find things in yourself that you'd like to fix, or maybe you don't? Maybe you're perfect.
No, but
I don't care. I don't care anymore.
my wife has a lot of those, by the way, but I wouldn't get into that.
A lot of things Dixon, you'd like to fix.
No, a lot of things you'd like to fix.
Dixon, you don't typically say so. It's very interesting. It's really good. Good. It's a good feature of yourself, Dickson.
Thank you. For
Origin of the MIMIVIRE system. So they do some phylogenetic analyses of these proteins, which are present in a number of MIMIViruses, and they group together and outside their bacterial homologs and other large DNA viruses. So they conclude, or they hypothesize, that these genes are present in the ancestors of these viruses. By the way, I never said what MIMIVIRE was, right?
Mimivirus resistance element.
MIMIVirus virophage resistance element, yeah. MIMIVIRE. I don't know. It doesn't quite slip off the tongue, but that's fine. They discovered it. They can name it, right?
Right.
So bacteria, let's think now. Bacteria in archaea are ancestral to eukaryotes, and this, of course, is a eukaryotic virus. So probably the bacterial archaeal CRISPRs were first, and then these came next, right? I should note that there has been one other report of a CRISPR system in a virus, and that is in a bacteriophage. And that was discovered by Andy Camilli at Tufts in Boston.
And Kim Seed.
Kim Seed.
Right. She's now a faculty member here.
She was in his lab?
Mm-hmm.
Oh, she's there. Cool.
Mm-hmm.
Neat.
In the MCDB department. Mm-hmm.
Nice. So we interviewed Andy on a TWiM. What was it? TWiM 56 was live at Denver. So check that out so it's in viruses as well. It's pretty cool. So that's basically it. Dixon, any comments? Thoughts?
Well, I had thoughts as I was reading this paper. Just
you had
in terms of, and I see you've read the email so that you can get to the lateral thinking part.
Oh, you read that?
Yeah, sure.
Go ahead, laterally think, Dickson.
Well, I tried while I was reading this, and the only thing I could come up with was imagining how these genes got stolen from the bacterial systems and then modified by the virus.
Why do they have to be stolen? Why couldn't they just slowly
covalent? You mean co-parallel evolution?
No, I mean we're all derived from a common ancestor, right?
Yeah, but how far back do you have to go for this one? I mean, this is like
the
primordial ooze.
One of the things about MIMIViruses is they're full of genes from a wide variety of different organisms.
Yeah, well, that's what I mean.
Which is consistent with, the way I think of it and the way it's been, I think, thought of by some others, is that if an amoeba or some other like organism is a normal host for these creatures, those organisms, eat lots of different stuff.
They do.
So there's exposure to DNA from a wide variety of organisms, which could facilitate this horizontal exchange of lots of different DNA. So there may be more opportunity for MIMIViruses than for lots of other creatures to pick up sequences from elsewhere, from bacteria, from archaea, anything the amoeba eats.
I think we shouldn't assume that MIMIVirus infection in its natural host is necessarily a bad thing for the natural host.
Yeah, no. In fact, there's some evidence that it's good.
So if this is a symbiont, getting back to Dickson's point, where did it get these CRISPR genes? If this is a symbiotic infection, and the organism that benefits from carrying the MIMIVirus is then susceptible to this phage infection that wipes out its symbiont, then that organism might evolve its CRISPR system, which of course it's had since archaea,
right?
And do so in a way that protects the MIMIVirus, and the MIMIVirus just has a habit of picking up stuff, and so this system that benefits it gets picked up and then becomes portable with the MIMIVirus.
Do you think that the amoeba hosts have CRISPRs that defend them against MIMIViruses and other giant viruses?
Wow, ought to have a look.
Yes.
The genome is done, I think, right, Dickson? Isn't there an amoebal genome?
Yeah, but I think that's for E. histolytica, Entamoeba histolytica. I'm not sure it's for the free-living. I guess you could look it up on GeneBank, but...
one of the things we just sort of, thinking about genome sequences, one of the things we just sort of cruised by in this is that if I understood it correctly, they did 45 new sequences of MIMIViruses for this thing.
Right.
Oh, yeah. We'll just sequence 45.
And by the way, this is a paper, this is a letter to Nature, which has, that genre has grown vastly. And there's one figure in the paper itself, and then there are, 6 in the supplemental data.
Absurd.
So the supplemental data is mostly what we've been talking about for the past 20 minutes.
It's ridiculous. It's absurd.
Yeah.
I would have been, frankly, happier to see more, shall we say, robust assays for the for the enzymes.
You didn't like those?
No, I wasn't crazy about it. I had to kind of stare at these and try and understand what they were calling nuclease activity. You can see some sub-bands in some cases and maybe some quantitative differences, but it wasn't too clear to me. And also, their helicase assay is more a winding rather than an unwinding assay.
Right.
And basically what they do is they incubate DNA with these proteins, and the DNA moves to the top of the gel. Well, I've had stuff on the top of my gels a lot, and usually I cut it off. Okay, we call it SOTT.
Schmutz.
Well, we have a different word for the S part on the top, right?
Yeah, that's great. SOTG, wonderful. That's great.
And, I would have liked to see controls like protease treating that stuff and making sure it's not the protein's just sticking to it and stuff. But I'm sure that this will be coming down the road. To me, the most compelling thing is the siRNA knockdowns.
Right.
Yes.
And the fact that and it's cool because the knockdowns actually increase the growth of virus on those. So it's not like you're killing the cell or anything like that. You're changing it in a fashion that actually allows something to happen. And I think it's really nice that these two Cas-like proteins are the ones which, when knocked down, give you more virophage growth. That's really very satisfying.
Rich, are you saying you're a KMVM kind of guy?
KMV Max. Yeah, I'm a KMV Max guy. Well, it kind of, hang around Niles for long enough and it kind of rubs off on you.
I just searched PubMed for CRISPR and amoeba, and there's nothing. Nothing.
Did you go to page 2? No.
Maybe I should try Google.
Sounds like Paul Harvey. Remember
Google
Paul
Scholar.
Harvey? Anybody here to remember him?
Yeah, I do.
Of course.
And as soon as he would pause, he said, page 2.
Yep.
So I wonder.
Look at this. Look at this.
What?
Now, Google Scholar, there are tons of articles.
Oh, my.
First one, the CRISPR-associated, this is Legionella, is required for intracellular infection of amoeba. Yeah, that's why you get multiple. Why didn't I get that in PubMed?
Very interesting.
I would have to go through these, but they look like all bacteria amoeba combinations. All right, so that is it for giant viruses. This episode of TWiV is also sponsored by the Microbe Magazine podcast. You should check this out. It's a new podcast from ASM. Goes along with Microbe Magazine, which is the monthly magazine you get if you are an ASM member. And the podcast is hosted by Jeff Fox, the current topics and features editor. Now, he's not the current editor. He's the current topics editor. You got that distinction, right? Each podcast coincides with an article in the current issue. And I've told you about episode 8 many times, which focuses on producing yeast strains to make lager beers with new flavor notes. Last time I had a beer, I didn't have any notes in it, but maybe I wasn't looking hard enough.
You shouldn't drop your notes in the beer anyway.
But the melody lingers on.
You know who used to say that?
Oh.
Harry Ginsberg used to say that here all the time, and he didn't play football at Duke, Dickson, or Michigan, or whatever you think
Right.
it is.
You didn't play football, period?
He didn't play it at Michigan.
He did play football because his knees were all messed up.
Oh, I thought he
and
did
later
play at
in
Michigan.
age,
I said Michigan.
You have said Michigan numerous times,
God.
and it's not Michigan.
And what is it?
it's Michigas.
It's Michigan.
Perhaps Duke.
Perhaps Duke. We could look him up. I guess we should Google him, too.
No, it wouldn't say, but I know he had both knees replaced later in life because he said he played football and he messed them up.
That's
and
right. That's right.
Our colleague Saul here also played football. What is this with virologists playing football?
No clue.
Anyway,
You get hit in the head enough times, you go into virology.
yeah.
I don't know what my excuse is.
You didn't play football, did you?
No, I didn't play football. You didn't either, did you?
No, I didn't play football. I played the real football. Soccer.
Oh, soccer.
Soccer.
We got an interesting letter. Let me finish the ad then.
Okay.
You can find Microbe Magazine podcast on iTunes by searching for Microbe Magazine, or you can go to microbeworld.org/MMP. We thank ASM for their support of TWiV. We got a great letter on TWiP from our friend Esper Callis, our physician in Brazil who we've heard from. And TWiP, we were talking about Wilson.
That's right.
Let's see if they get that, Dickson. Wilson! Remember from that movie?
Yeah.
With Tom Hanks.
Tom Hanks.
So Dickson said it was a basketball, and Esper wrote in and said, "You guys,
Oh man!
you guys are you made such a huge mistake. It's a volleyball."
Yeah.
He said, "I know this because I recognize this ball because this sport is played internationally as opposed to weird things you play in the U.S."
But then I said, guess why it was named Wilson?
Well, that's
Because the ball was made here.
Yes, by the Wilson.
Yeah, it's a Wilson company, right? It was Wilson. There was a label on the ball. Right?
We know about soccer.
Now,
We do know about
living as I do right in the Springfield Holyoke area, I can tell a basketball from a volleyball.
That's right.
So.
He actually sent us a picture.
He did.
Okay, I just put one in too.
Remember, he had put the straw coming out of the top or something, right? Yeah. Okay, we have a few letters. Kathy, can you take that first one, please?
Sure. Phoebe writes, Dear Vincent and friends, I am a professor at a small liberal arts college where I teach and do research with undergraduates. My PhD is in microbiology and molecular genetics. Although there were virologists in the department, I earned my PhD studying bacteria rather than viruses. Through teaching undergraduates a senior-level elective in virology, I've noticed that the term reversivirus, referring to double-strand DNA reverse transcribing viruses, is found in many online sites that undergraduates like, for example, Quizlet.com, Slideshare.com, Wikipedia, and quite a few others. My students are intrigued by this evocative word, reversivirus. But if I search the vast web of knowledge database for the term reversivirus, I get no hits at all. I can find reversivirus used in quotations in a few books using Google. One of these references is as early as 1988, but at that time the term pararetrovirus was sometimes also in use to refer to this same group. I can find the term in some textbooks, such as the book Topley and Wilson's Microbiology and Microbial Infections, and the book Principles of Microbiology by Sumval and Mahrota, but not in others, yours, and Fields. I'm a microbiologist, but not a virologist. Thus, I do not know if virologists actually use the word reversivirus in casual conversation or in teaching. I did notice that TWiV number 49 calls these gapped double-strand DNA genome viruses.
Aha.
What do you think? Is reversivirus a wonderful evocative term for double-strand DNA reverse transcribing viruses? Or should I throw it in the file 13 and caution my students against its use? Do you know who may have invented it? Thanks, Phoebe.
This reminds me of Star Trek. I'm a microbiologist, not a virologist.
That's right. Damn it, Jim.
I vote for file 13.
Yeah, I do.
I looked it up on Google Ngrams, and there's nothing there all the way up through 2015. So
Has anyone
I don't
ever heard of it?
I don't think I've seen it until this letter.
Yeah, me too.
I'd never seen it.
Huh.
So it sounds like she's referring to the hepadnaviruses, right?
Yes. Yeah.
Right.
So Anne Skalka, in the latest Principles of Virology, she calls all of these the hepadnas and the cauliflower mosaic virus, any virus that has reverse transcriptase in the reproductive cycle. She calls them retroid viruses. Retroid, which I'm not terribly fond of, but yeah, I never heard of reverse E virus. It's weird. I don't think David Baltimore or Howard Temin would like it either.
So you can cure an infection, but you can't reverse a virus.
Oh. Yeah.
For some reason recently, I was going back and looking at really old, I think, parvovirus papers, and there was some other name that used to be used for parvoviruses. I can't remember what that was, but it really...
Not the pendoviruses, right?
No. It was, something way more obscure than that. It was and really old. I should have written it down. Oh, well.
Rich, can you take the next one?
Sure. Fritz writes, good morning. The the requisite weather report. It is 26 Fahrenheit or -4 C and relatively clear on this early March morning in northeast Pennsylvania. I really wish I knew my metric units better. Had to look it up to give this report. No sweat. I still have to use my little converter every time. This is a bit pedantic, but I know you have been taught not to anthropomorphize.
Right.
In TWiV 372, you mentioned that the virus evolution takes something into account. It seems that taking something into account is anthropomorphic in nature. Not sure what wording could be used instead, but I thought I'd point it out. As mentioned in the episode, sometimes anthropomorphism helps, even if it's not technically correct. Love the show. Learning a lot for someone not in the field of virology.
Virus evolution takes your critique to heart.
Yeah.
No, he's absolutely right. Or she, and it's sometimes hard to avoid it. Yeah.
Yes. It is very useful in terms of explaining what's going on in many cases. I understand why in a textbook you don't want to do it, but I think in conversation it's sometimes.
Let's ask Carl Zimmer next week.
Oh.
Yes.
Ooh, good idea.
We have to write some questions down.
That was my big secret. I didn't know if we were letting it out that he was going to be on next.
Oh yeah.
And
I was wondering if he says so when he starts sentences. Let's find out.
We are going to find out.
The last one is from Karina, who writes, thank you for bringing the oft-overlooked legacy of this pioneering, preeminent, world-renowned virologist to light. Thank you. Thank you very much. Oh, not me. As a graduate student in Dr. Youngner's department, we were in awe of having such an unassuming luminary in our midst. Historic accounts of the development of the polio vaccine have largely focused solely on Dr. Salk and omitted the seminal contributions of this great scientist of immense integrity. Is the talk you gave available online, Dr. Racaniello? Another couple of recommendations for future visits to Pittsburgh, Joe Glorioso and Charles Rinaldo. Thank you for all your contributions to virology and science and on YouTube. I'm not going to Pittsburgh in any winter ever again. I will go in the spring, summer, or fall. But yeah, Joe would be great. Yeah.
So is your talk available online?
You know, it was recorded and I have it, so I just have to edit it and put it up. And I thank Karina for reminding me. Yeah, it was a fun episode. Youngner's great. He's 94, completely with it. And he gave me a hug when we left. It was really nice. And the last thing I want to mention is I was cleaning out my office today. Last week, and I threw out all my journals of virology, all of them. I recycled them. I saved one, the very first journal I ever bought. It is March 1973, volume 11, number 3. It's about 1/4 of an inch thick. And listen to this editorial board, which is hardly anyone, but this is such a who's who. The editor-in-chief is Robert Wagner. Myron Levine from Michigan was an editor, and Norman Salzman was an editor. And then some of the associate editors, let's see, David Baltimore, Marcel Baluda, Alan Campbell, Pernell Chopin, Vittorio Defendi, Walter Dorfler, Richard Franklin, Harry Ginsberg, Mark Girard, Angus Graham, Paul Kaysberg, Albert Kaplan, Ed Kilbourne, David Kingsbury, Lloyd Kozlov, Margaret Lieb, Jean Lindenman, one of the discoverers of interferon, Chris Matthews, Bernie Moss, Elmer Pfefferkorn, Leonard Phillipson, Fred Rapp, Bernie Roisman, Aaron Chatkin, Robert Simpson, Don Summers, Howard Temin, Peter Vogt, and Julius Youngner.
Wow.
Nice.
Isn't that cool?
Very cool.
That
Yeah,
is
that's
cool.
very
Yeah.
cool.
So many really well-known names. And today the journal is really thick, and there are lots of editors. And I haven't seen a paper copy in ages.
They stopped making the paper copy last
Is
year.
that right?
They did. Wow. And there were just a few articles here, and I would say half of them, more than half, are on bacterial viruses. So they used to be phages here before they took them out and put them in, Journal of Bacteriology, right? I think so.
Wow.
Well, this is so cool. Anyway, I digress. Science Picks of the Week. Alan, what do you have?
I have an infographic, but it's not the kind of infographic you glance at and say, "Oh, I get it," and then go on to other things.
No, I was having a little trouble with that.
This, you have to click on it to get the image by itself, and then you have to click on it again, and you actually have to download it in order to be able to read all the text on this thing. It should be, ideally, it should be blown up to about wall mural size.
Yeah.
Wow, it is big.
So if you save the image, I think that's what I did. I think I saved the image and then opened it separately on my computer so that I could bring it up and look at it. But it is a graphic that traces the evolution of thought about evolution.
It's all pixelated though when I do that. Can't read.
Gosh, I gotta.
You have to save the PDF.
That's true.
I did, but it's still, when I zoom in, it's pixelated.
In download, if you click the link that says download
Maybe it's a higher res.
and download the highest res one
okay,
which is
got it.
950K.
Yes, there you go.
That's what I did.
They're different versions.
Oh, I just downloaded it in French, but okay, you'll like that one. Yes, that one blows up and is readable, but it goes
Wow.
way back, and it's got a timeline underneath, and it traces. Obviously Darwin is a huge player in this, but it's interesting to see. He's certainly not the only one. There's a lot going on back to the 4th century BCE, and thinking about where different species come from and why there are so many of them, and different theories that didn't make it, and on and on.
Yeah.
Lovely.
So it's got lines that connect all these, which I like.
Yes.
And what I like is, just as an example, in the 18th and 19th century, it's got a little box about Lamarck, who's talking about the notion that characteristics are acquired because an animal requires something of his environment, like giraffes acquire long necks because they got to reach for trees, and says, but that was wrong. And then there's a little box that says, or was he? Or was he wrong? Okay.
And then it goes
and an
forward.
arrow going all the way to the present where they're talking about epigenetics.
Yes.
I like that.
Yes.
Good note. So
Like-
downloading is the key, Alan. You're absolutely
Yes, you
right.
have to.
Yeah.
Yeah, I should mention that.
I like
you
the quote
should,
though, from Dobzhansky too.
You should download the full resolution version in order to read the little boxes.
Nothing in biology makes sense except in the light of Despommier,
No. That is not correct.
in the light of evolution.
Yeah.
Oh, this is lovely.
He was a genius.
And they include systems biology here at the end.
Where does Wallace's box in that
Next to Grommet, I would assume.
arg?
Dickson, do you know what he meant?
A little bit.
Wallace and Gromit. Oh, you should watch that series of it's a series
Oh my
of movies
goodness,
by animated movies by Nick Park.
Yeah. Stop motion animation. That's
Yes.
fabulous.
Okay.
Fun.
Dixon, do you have a pick?
I do. I thought I sent it to you.
It's part of the show. I'm asking you.
Yes, I have a pick. It's about vaccine deniers, and it's a big tome about what happens to certain children that are not vaccinated, and it's really quite riveting to see that still there are people out there that deny the need for taking advantage of modern medicine in its fullest.
Yeah, there's a story of a young child who died of meningitis, and his parents didn't take care of him properly.
Exactly.
There's a lot of controversy over this. I found a really good summary over at Science-Based Medicine of this whole case. It goes on and on. It's very long. And
Science-based anything is taking a hit.
yeah, this is really
Taking a
sad.
bad hit. It's a very sad
Yes.
story.
Yeah, so they apparently they took him to various naturopathic this and that and the other, and had him, fed garlic or whatever.
Yeah, that's right. Garlic
was being peddled, and
he wasn't immunized either.
He wasn't immunized against anything. They were all into natural everything, and the kid ended up dying as a result of this parental malpractice, which is essentially what the parents are being charged with now. They're criminal charges.
Do you know there's a book by Gary Larson called "There's a Hair in My Dirt"? You know that book?
No, I don't.
Oh, that's my second pick.
The title is great.
There's a hair in my dirt, and it's about people who deny the
HAIR,
HAIR, hair in my dirt. And the whole gist of the book is that people think that life is soft and fuzzy, except that it's pretty harsh. And this hair comes from a little girl who died and then gets consumed by a family of worms. And the family of worms, of course, the little kid in there sitting at the dinner table saying, "There's a hair in my dirt." And it's one of the golden hairs from this little girl who died as a result of evolutionary process. As you will see, Gary Larson was very good at accuracy of biology. But it was prevalent, I don't know, is this denier issue of Alan's first pick. I mean, you should have had divided it into two parts, creationists and non-creationists, and then shown the evolution of thought. I bet you there's some crossovers in both of those things.
Right.
So that's very depressing to realize as well, because no matter how logical and straightforward you appear to be to everybody, you're not always perceived that way by everybody. And they can find plenty of holes in what you're saying to back up their own point of view. And this little kid is the poster child for that. And that's what happens when things go wrong. That's why I picked it.
Thank you, Dickson.
You're welcome.
Rich, what do you have?
I have something that I just kind of ran across. Where is it? It's getting buried in all this stuff here. It is Physics Girl.
Yes.
This is it's both a website and a YouTube site. I've linked the website here. Let me read. I found it because of this one YouTube clip that I noticed somewhere on Crazy Pool Vortex, where she runs a plate through the surface of the water on a pool, and it creates these twin vortices that then march across the pool. And she has a whole big rap on why this behaves the way it does, and she's very, very clear and very enthusiastic. And this, she's an amazing woman. Let me see. Her name is Diana Cowern. She's the creator of Physics Girl. I'm reading from her about thing here. Yeah. Driving tractors on a farm in Hawaii where she grew up somehow led to science and engineering pursuits. Nicknamed Happy Pants in college, and if you watch her videos, you can understand this. Diana researched dark matter with Professor Joycelyn Monroe as an undergraduate at MIT, and low metallicity stars with Professor Anna Friebel as a post-baccalaureate research fellow at the Harvard-Smithsonian Center for Astrophysics before bringing her happy intern pants to GE, where she worked as a software engineer designing mobile apps. Eventually, she started exercising the nerdy side of her brain on YouTube as Physics Girl before and while heading over to UCSD as a science outreach coordinator. She loves to surf, scuba dive, and play the ukulele. And these videos of hers are fantastic.
She is an extremely good science explainer. This is
absolutely.
I just watched this Crazy Pool Vortex video when I was looking through the show notes before the show, and I thought, wow, this is outstanding.
Yeah, she's very good.
She's got not only the explanation, which is pitched perfectly for anybody to understand, but the enthusiasm behind it is just. It's infectious, and it's so representative of how scientists at their best approach things.
Great.
Happy Pants.
Cool.
Happy Pants, yes.
I'll have to send her.
And she's got a ton of them.
Yeah, it's good. UCSD, that's great. It was out there not too long ago. Kathy, what do you have?
I picked two things that I found from a blog that I read. It's ecology and I guess, somewhat evolution blog. And one is the Noun Project, and the other is PhyloPic. And the Noun Project has, in the search bar, you can just put, I guess I linked to the general page. You can just put in a word, like you can put in virus, and then follow it through to get a bunch of basically silhouette black and white icons of some viruses. And you can be a member for free and purchase royalty-free icons for $1.99, or you can be a Noun Pro for $10 a month. So I thought this might be useful to some people. And then the second one is more of a biology-based thing. I'm trying to get back to it now. The PhyloPic. And there you can, again, it's black and white silhouette icons of various animals and things. And I tried putting in virus, and it seemed hopeful, like there were a bunch of sub-links, but then I really never got to anything. But if you put in, for instance, elephant in the search bar, then you get some things that are really elephants, and you get other things that have elephant in their name. And you can follow that around. And that everything seems to be, it's free silhouette images of animals, plants, and other life forms available for reuse under public domain or Creative Commons license. So 2 different ways to get some black and white icons of things.
Kathy, we typed out the word elephant, and all the words that came up looked truncated. Sorry.
Very good, Dickson.
If you search for virus and you scroll down far enough, you find what looks like an adenovirus.
Okay.
I don't know if you found that.
You might have to go to the second page.
Right.
You might have to go to the second page.
Yeah.
They have an icon of a pregnant woman and what looks like a virus. Yes, it says Zika infection. They already have a Zika.
Really?
Yeah, you hover over it and it says Zika infection. So it's a pregnant silhouette and there's a virus hovering over her big belly and it says Zika infection. Wow, they are up to date.
So when you do the taxon search and you get viruses, then you get a bunch of these blue things. And so then where did you go from there?
I typed in viruses and just hit return. Or virus, sorry, VIRUS, and then I hit return, and then when I scrolled down quite far down the page, there was an adenovirus. You just have to keep scrolling. Yeah, there it is.
We must be on different pages because I get this box that's called taxon search.
Right.
Yeah.
Yeah,
I get that. I get the
that.
taxon search, and then you can get arenavirus 7.
Even I can see where Ebola is.
Yeah, no, I don't get. I get ones that are all virus. It's just virus and hit return. I don't do any taxon search.
But there's some ticks in there. There's
No, I
vector
didn't choose to
pictures.
either. It just brings it up.
I didn't get that. It's interesting. Anyway, there's
Okay, so it takes a little more exploring.
Right.
Boy, that's pretty neat. I like icons. I like the one that it's a guy's face and a mosquito biting on his nose. It's very cute. And there's a lot of good stuff here. Could have a lot of fun with that. All right, my pick is a post from Science-Based Medicine. It is called "The Claim That Gardasil Causes Premature Ovarian Failure, Ideology, Not Science." Now, Science-Based Medicine is always very good, and this is addressing the HPV vaccine, of course.
Yes.
And it's really unfortunate that people are speaking out against it. So this is a great article that debunks all of that. And this one paragraph I'd like to read. So what makes the conspiracy theories about Gardasil different? Simple. Anti-vaccinationists have a special hatred for Gardasil. I realize that it sometimes annoys some of our readers when one of us waxes political or ideological, but in this case, it really can't be avoided. The hatred and fear of Gardasil and Cervarix very much appear to derive from the fact that HPV vaccines are used to prevent a sexually transmitted virus, the rationale for that fear being that the use of such a vaccine will somehow encourage promiscuity. It's even been called the promiscuity vaccine. Never mind that the evidence is quite clear that this claim is simply not true. Never mind that anyone who remembers her own adolescence clearly would know that fear of catching HPV and then developing cervical cancer 20 or 30 years down the road is not a major concern among teens as their raging hormones drive their behavior. None of this matters to the people making these claims, however, who seem to think that cervical cancer is just as a just punishment for promiscuity and naturally seem to latch on to conspiracy theories in which HPV vaccines would naturally cause infertility because the promiscuous shouldn't be mothers. Really good article. You should read it. But unfortunately, the people who need to read it will not. Right?
Yep.
Well,
I have to really kind of count to 10 when
I must tell you that this harkens back to an old Greek myth of Pandora's box, which I've referred to earlier in several of the shows, where the diseases of the world are blamed on curiosity of some woman who decides to open this box against the will of the gods and releases all of the infectious agents. That's the origin for a lot of this creationist illogic, I should say, and the punishment quotient of if you catch a disease, it's because God wants you to be sick. That's just an impossible thing to get out of the mind of a patient that comes into a hospital who's got that as their life attitude. And doctors have to deal with that. They refuse therapy because of that. They see it doesn't do any good because God has willed this. And I've heard those stories from medical students that have gone onto the wards and had to interview patients. And you dig deep enough, and you can find a lot of that in the world.
Bottom line: the HPV vaccine is effective and safe.
Yeah,
that's an article by David Gorski, by the way. We also have
remarkably effective and remarkably safe.
Yes,
it's amazing.
Yep. We have a listener pick from Mark, who was our guest in studio a couple weeks ago.
Oh, right.
He writes, hello, Alan, Dickson, Kathy, Rich, and Vincent, or those who are present. Mm-hmm. As I write this in early March, I am sure the weather where each of you dwell is somewhere between wonderful, terrible, or just meh. Today here in San Jose, California, we had a mostly cloudy day, temperature in the mid-60s in between rain days in our current 7 to 10 day El Nino storm pattern. We discovered it in January at the start of California's current El Nino. For those keeping track, our state is about 10 to 15% ahead of normal rainfall.
You need it.
This is good news, but not enough to recharge depleted reservoirs.
Exactly.
And water tables.
Exactly.
To TWiV listeners, to learn more about predicted and current weather, I recommend MyRadar, an app available on iOS, Android, and maybe other smartphones. MyRadar uses data from NOAA, the National Oceanic and Atmospheric Administration. The choice of display colors for maps and overlays lets you customize something as common or idiosyncratic as you desire. The MyRadar app lets you view current weather conditions. Viewing storm fronts is especially valuable. You can pick a location and center it on the map with a tap on the screen. Then you can adjust the magnification and zoom and see what weather is coming where you are or want to be. It shows rain, snow, or other precipitation, or simple sunshine. This may sound confusing, but it is really simple if you download and use this free app. You can also overlay wind directions, a cool vector field for all of us who vaguely remember their vector calculus. Well, I don't. Note that the free version has a small ad, which an in-app purchase upgrade will remove. Other informational displays are available for in-app purchase as well. It sounds cool. I have to get one.
Yeah.
Can never have too many mail programs, right? Not mail, weather.
Weather.
What do I care about what mail programs?
I have no idea.
Mail.
Because this came as a letter.
Thank you, Mark. And that is TWiV 380. You can find it on iTunes and also at microbe.tv/TWiV. And do send us your questions and comments to twiv@microbe.tv. Dickson Despommier can be found at Racaniello.org, which we found the other day.
We did.
Is a remarkably useful site.
Oh.
Thank you, Dickson.
You're welcome.
That was on TWiP.
Yeah.
You remember?
I do.
And you need to be nice to Dickson, okay?
I'll try.
Are you done?
No, but it reminded me of a stupid little saying that says, roses are red, violets are blue. I'm schizophrenic, and so am I.
I never heard that one before. Very good. Thank you, Dickson.
You're welcome.
Kathy Spindler, sorry, let me do that again. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
It's not hard to pronounce your name. I don't know why I did that. Sometimes your tongue gets tied. Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
And Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Thanks. Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.